Hi everyone, my name is John Michael Sauer, and I work at the Critical Path Institute. I'm actually a program officer there. I'm the program officer for the Translational and Safety Sciences program. And today what I'd like to do is spend a little bit of time and talk about the outcomes for our Crohn's disease biomarker pre-consortia, which was a pre-consortia that was funded by the Helmsley Charitable Trust. Let me go ahead and cut right to the chase and really talk about what the primary accomplishments were within the consortia or the pre-consortia. And that was this publication you see in front of you, Biomarkers of Crohn's Disease to Support the Development of New Therapeutic Interventions. And as described by the title, that's exactly what we go into, is we talk about very specific biomarkers. We look at their regulatory as well as their biological relevance. We also go ahead and look at the quality and availability of the biomarker assays for each of these biomarkers. And then we also took an in-depth view in looking at the current clinical validation of the biomarkers. So, what is the evidence that's been generated around these various biomarkers for them to be used in drug development as well as in clinical practice. You can also find some additional information that we put together on the c-path website. And what this did was very specifically outline our landscape assessment that we did, and all of the information that we gathered around various biomarkers, their available data, literature associated with them, stakeholders that are interested in these biomarkers are actually found in that first PDF you see there, the landscape assessment of Crohn's disease fluid biomarkers. The other two documents you see on this web page are actually a regulatory readiness checklist for both calprotectin, fecal calprotectin, and serum C-reactive protein. And what these documents include are really in-depth evaluation of both of these biomarkers and how they could be applied within Crohn's disease. But why don't we take a step back and see where we started? So when we first started the pre-consortia, what we really wanted to do was have a broader understanding of the world of Crohn's disease fluid biomarkers to create a landscape, and then to really define a roadmap for how we could initiate a consortium that would be focused on the qualification or regulatory acceptance of these biomarkers for use within drug development. And very much as I described about the paper, what we did was we looked at clinical validity, assay validity, as well as the availability of patient-level clinical data, because that's required for what we need to do within the realm of regulatory qualification and convincing regulators that we have biomarkers that do what we say they do. What we did was we pulled together a number of stakeholders, academic stakeholders as well as industry stakeholders. And what c-path did was went ahead and coordinated this expert team to go ahead and basically do the heavy lifting around evaluating these various biomarkers. So let me tell you a little bit about biomarker qualification beyond what I've already told you. It's actually a formal regulatory review process that results in the acceptance of biomarkers for their use in drug development. This is really important for drug development sponsors, because drug development sponsors want to make sure that they'll understand how the regulators, the health authorities, are going to react to any data generated using these biomarkers. And so what it does is it creates basically a blueprint for how these biomarkers can actually be used with all stakeholders, right? Industry is involved, academia is involved, and so are the health authorities. Now, one important thing to remember is that, even though qualification doesn't denote that this biomarker can be used in clinical practice, these very same biomarkers could be used in clinical practice. That'd be a different route to basically receive endorsement around clinical practice biomarkers, but the data set that would be generated is the exact same data set. And so we're really doing two things at once here. Even though we're focused on drug development, we also have clinical practice in mind. So how do we do this? What was our strategy behind the pre-consortia? It was really, number one, identifying the drug development need, right? What's the gap? What needs to be solved? Where don't I have a tool in drug development that I need a tool? We then aligned these needs with specific regulatory needs to make sure that we're not only speaking the drug development sponsor language, but we're also making sure that we're speaking to the health authorities as well, right? And we're aligned around how we're going to have the conversation about why this biomarker is going to overcome this gap and how it fits into the regulatory process. We then went about identifying single biomarkers and biomarker panels to support the need, right? Those are going to be the potential tools that can overcome that need or that gap. Then what we did was we created those documents that you saw on a previous slide, right? These regulatory readiness checklists to make sure that the biomarker fit and was ready to actually be able to be pursued for regulatory qualification. Like I said before, one of the really important parts was making sure the data's there, right? Because decisions made by regulators are, of course, data-driven. And so you need to basically work with them and show those regulators that data as you go through the qualification process. One of the final parts of the strategy was really making sure that the qualification strategy that's been derived with stakeholders really fits with what health authorities think should be the strategy as well. And so this is really kind of the formal, informal meeting, if you will, with regulators to go ahead and make sure that everyone's aligned with how we can move forward with receiving endorsement around these biomarkers. Well, one of the main things that we also did was, we prepared for the full consortium, its formation, right? We put together a project plan, membership agreements, and we had discussions with various potential members. Our ultimate goal was then to really launch the consortium. Well, we haven't done that yet, right? And I think COVID-19 and the pandemic got in our way, but we're still very much interested in that. And that's one of the reasons why, I'm presenting this information to you today for your consideration on whether or not you would be interested in participating in such a consortium. Within the pre-consortia, what we did was we formed two task force, one that really looked at predictive, prognostic, and treatment option type biomarkers. And the other group looked at disease activity biomarkers. What we did was we had two industry members who helped lead, Yuri Albrecht and Josh Freeman, as well as two academic members, Ted Denson and John Braun, to lead these groups. It was actually pretty funny because as we began to have conversations, basically these two groups began to converge, which was a good thing because I think what it did was it allowed us to then identify these biomarkers that had multiple uses, and we could come up with more of a longitudinal strategy of how we could work through the qualification of these biomarkers. In each case, what we did was we came up with a particular context of use around how the biomarkers could target these gaps. The teams worked to populate the biomarker checklist from literature as well as from internal knowledge from both academic investigators as well as from industry investigators. Biomarkers were then stratified by plausibility and their likelihood of success, as well as, importantly, the availability of supportive data. And so what we basically did was, as I said, we worked through the different potential gaps, and you can see those on the first bullet point, where to identify these high value blood and fecal Crohn's disease biomarkers. Some of the examples looking at, patient selection, right? A prognostic biomarker that could identify a patient population that would progress into the next stage of the disease. Biomarkers that could look at disease activity, mucosal healing, disease complications. And then we got into biomarkers that were potentially even more valuable. Those that actually would look at treatment response, pharmacodynamic biomarkers, as well as those that could actually stratify treatment options. And so those could be extremely valuable within the drug development arena. And so, as I said, this was basically what we went through, the unmet need, the specific regulatory need, as I showed you on that flow scheme, the availability of clinical data, the utility across multiple drug classes, because that's a really important thing too. We don't want to have biomarkers generally that just are going to support one drug class, that are mechanism-based biomarkers. That's really not going to do the community as much good. Sometimes those biomarkers are valuable to pursue, but in this case, they were set at a lower priority because of the goals of the pre-consortia, and then of course the regulatory readiness strategy. So here goes an example on the left of this regulatory ready document and what it went through. Well, one of the things that we noticed during our venture through the landscape of Crohn's disease biomarkers is that there really weren't a lot of regulatory ready biomarkers. And so what we did was we looked at discovery or exploratory biomarkers as well. And that's also included within the manuscript that I previously showed you. And I think this is really where the excitement is around these exploratory biomarkers. But I don't think we should walk away from these shall we say, regulatory ready biomarkers either. Let me show you how we went through this in a tabular form here. As I said, we broke these biomarkers up into different gaps, and we then broke them up into the different biomarker classes, right? Pharmacodynamic, prognostic, predictive biomarkers. And then we had generalized context of use statements that were associated with each of these gaps. And what we did was, where we saw a lot of data, we basically then ranked these biomarkers, right? And we then did a deeper evaluation. And what we really found were there were three major biomarkers that kind of rose above all the other biomarkers, and that was fecal calprotectin, which is basically a product from neutrophils, right after they activate and they basically lyse themselves. Calprotectin is released into the GI tract, ends up in the feces, and then is excreted. Likewise, fecal lactoferrin, also a neutrophil product, is also released into the feces after neutrophils activate. Interestingly, serum C-reactive protein is also, at least from a pharmacodynamic standpoint, a biomarker of interest that there's significant data around. You can see there's some of the cytokines that received a couple points. But then what we do is we begin to move into an area where there is less and less clinical validation around these biomarkers, and less data that's available for their qualification. And so what that really resulted in is then a panel of exploratory biomarkers where we could see that they were being used, but what we'd have to do is, in order to take them forward for qualification, we would need to go ahead and generate additional data. And so what you'll find in that online PDF, as well as in the paper, are the listing of these biomarkers and all the information that we generated around them and put together from the literature, as well as from conversations with academic and industry investigators. And as I said before, folks are really excited about the exploratory biomarkers just simply because they're new and exciting. And so what we really found is, at the end of the day, and we're getting towards the summary here for what the pre-consortia did, is, fecal calprotectin and serum C-reactive protein, or CRP, were identified as regulatory ready biomarkers. You might ask what happened with lactoferrin. Well, basically, it correlates exactly with fecal calprotectin. It just turns out that fecal calprotectin is easier to measure. And so, if we were going to go after to qualify these biomarkers. It would really be fecal calprotectin that we would pursue. Likewise, when we looked at the exploratory biomarkers, there's a significant amount of interest, but that's going to take additional work to really figure out what needs to be done and how we would put those biomarkers on a trajectory for, prognostic or pharmacodynamic or predictive biomarkers. One thing that we didn't do, but we're currently in the midst of doing, is looking at ulcerative colitis. As I'm sure everyone knows who's listening to this, there's two major diseases within inflammatory bowel disease, and that is Crohn's disease and ulcerative colitis. There's some sub-diseases, but what we're going to do is we're going to also look at UC and see what biomarkers we can go with there. It turns out a lot of those are going to be similar to what we're looking with Crohn's disease as well. So there's going to be a great complementary overlap. And I think what the bottom line is after all of our conversations, what we've come up with is speaking with industry members as well as speaking with regulators, is really looking at fecal calprotectin and serum CRP for Crohn's disease as a pharmacodynamic or response biomarker. And I can get a little bit into that in a moment. But one of the things that we did was we had some really good conversations with the regulators, around how we need to move forward. And the dates aren't important here, but what the important part is that we were actually, at the FDA, with the review division, able to really figure out where the biggest impact would be. And it's exactly what I talked about before. It was around the pharmacodynamic biomarkers. Now, so what we would like to do is we'd like to move in and create IBD risk, or the Inflammatory Bowel Disease Regulatory Science Consortium. And what this consortium would do is basically take what the pre-consortia did and actually utilize that to go ahead and then work towards the qualification of biomarkers to reduce the uncertainty and accelerate drug development of therapies for the treatments of Crohn's disease and ulcerative colitis, or IBD. Now, when we imagine what this consortium would look like, it's pretty clear that what we would need to do is we would need to have basically a regulatory ready work stream and then an exploratory biomarker work stream. I think that would make a lot of sense because we're very quickly going to run out of regulatory ready biomarkers, and we'd have to, when we want to bring forward these exploratory biomarkers to fill in the gaps that those regulatory ready ones just simply do not fill in within Crohn's disease or ulcerative colitis. One of the strategies that we could use that we've done very successfully, at c-path, is basically figuring out quantitative solutions to build tools that will allow us to best describe the utility of the biomarkers, but then also be able to use the biomarkers more simply in clinical trials and drug discovery. So, getting back to the story of the regulatory ready biomarkers is and the reason why I'm proposing this is because this is actually something that we could do today. All the data is available. It's out there. We know it is, right? But there just hasn't been the push right now from industry to go ahead and pursue this. But one of the big gaps out there is the lack of biomarkers that can determine patient response to new therapies beyond ileocolonoscopy. As we know, ileocolonoscopy is an invasive technique. I mean, it's not a desired approach that patients like either. What we would propose in a context of use statement is that fecal calprotectin and serum C-reactive protein are both pharmacodynamic response biomarkers that are able to identify patients with Crohn's disease that have achieved endoscopic healing. I think we can do the same thing minus CRP for ulcerative colitis as well, and categorize these individuals as being in remission during the conduct of phase 2-3 trials. So what does this really save us? Well, it's going to save us the number of ileocolonoscopy procedures that would be required to demonstrate effectiveness of a therapy. We can think about this in an adult population. The other thing is we can also turn it around a little bit and think about this in a pediatric population where sometimes ileocolonoscopy is not an option. But ultimately, the idea would be is to correlate endoscopic healing as evaluated by ileocolonoscopy with the lowering of both fecal protectin and serum C-reactive protein. So within a nutshell, what we could very quickly do is move to a qualification or regulatory acceptance of these biomarkers and really standardize the way that regulators are going to look at them. Right now, regulators accept CRP as being able to identify an individual with IBD, but really, they don't accept these as true clinical trial endpoints. Ileocolonoscopy remains that hard endpoint for IBD trials. So with that, I'd like to thank you. I appreciate your time, and I really hope to see you at the webinar during the live sessions where I'll gladly take any questions around what I've presented, and look forward to seeing everybody. Thank you.